Welcome to Human Physiology, Chapter 2, The Cell, Structure and Function. This particular audio is going to be no longer than an hour and 15 minutes, so if we do not finish this in that time frame, you will need to look for a second audio. Alright, first of all, let's start off with what is a biomolecule. A biomolecule is any molecule synthesized by a living thing. And with Earth, and humans specifically, this is typically going to contain a carbon. There are a couple of bacteria that create molecules that don't have carbons, but by and large, they are going to contain carbon. Now, for our purposes, we classify them into 4 categories. If you took general biology with us, these are going to sound familiar. So this should be a review for you. These 4 categories are going to be our carbohydrates, our lipids, our amino acids, and proteins, and then our nucleotides. Now, 3 of these, even if you haven't taken Gen Bio here at Verse State, should sound familiar because they're essentially your 3 macros for nutrition: your carbs, your fats, your proteins. The 4th one we don't talk about nearly as much as a macromolecule, but any conversation we have about DNA, RNA, genetics, we've talked about nucleotides. So let's start off with our carbohydrates. Our carbohydrates are going to be broken down, or referred to, I should say, as saccharides. These saccharides are going to be at their base form known as a monosaccharide, singular. This monosaccharide is also referred to as the carbohydrate monomer. And we have 3 main types of monosaccharides, and they all share the same chemical formula. So when I just write out how many carbons, hydrogens, and oxygens are in these monosaccharides, they are going to look the same. There's nothing telling me it's a glucose versus a fructose or a galactose from just the chemical formula. That chemical formula is C6H12O6. 6 carbons, 12 hydrogens, 6 oxygens. And what you see below here is their actual formation. Now, this is a 2D model of a 3D object, but this is how those carbons, hydrogens, and oxygens are actually linked together. And you can see some of the differences are where's these hydroxyl groups. This OH is a hydroxyl group. Where is it? What carbon number is it attached to? It's attached to carbon 1, 2, 3, 4, 5, or 6. In the case of fructose, it is actually a pentagram, not a hexagon. Glucose and galactose are both hexagons. So some of that's the number of sides on the ring that they make, what number are the hydroxyl groups on, all sorts of things. So this is what makes them different, but they all have the same number of carbons, hydrogens, and oxygens. These hydroxyl groups, these OHs, actually make these carbohydrates polar. Now, again, hopefully that sounds vaguely familiar from our conversations about bonds in chemistry and general biology. Covalent bonds are bonds where the atoms share electrons, and a nonpolar bond, remember, is when they share them equally between the 2 atoms, and a polar bond, it is going to be shared unequally between the 2 atoms. So these hydroxyl groups are going to have an uneven sharing of electrons. Now, these monomers are going to link together to form disaccharides. So disaccharides, di for my med term students, remember that means 2. So 2 sugars. And how they link together, a beta link versus an alpha link, which we're not going to get into, but how they link together is actually going to determine what type of disaccharide they are, as well as what 2 monomers are linking together. For instance, in order to get lactose, the bane of many people's existence. Lactose is the sugar that many people who are lactose intolerant are unable to break down, and it is what causes them to have a bad relationship with dairy. That's this guy right here. Lactose is simply a galactose and a glucose monomer linked together. Whereas sucrose, our good old table sugar, the thing we love in the form of our candy and our sweets, our like brownies and cakes and stuff like that, that's simply a glucose and a fructose linked together. And then we can link all of these monomers together into chains and branching chains to form polysaccharides. Again, poly for my med-term students, hopefully you remember that means many. And so many sugars. This right here is an example of a glycogen polysaccharide. It is many glucose monomers linked together, and it links in a branching format. There's lots of different polysaccharides. Fiber is going to be one of them, all the different types of fiber. Those are all branching carbohydrate chains. Now, to make polysaccharides or disaccharides, or to break them apart, we need to talk about covalent bonds, because that is going to be what links these saccharides together. It is a covalent bond. Now, when we have covalent bonds, if we're going to join them together, that is going to be a condensation reaction. In other words, we're going to remove a water molecule, an H2O, from these two monomers, and that removal of a water molecule is what's going to link them together. If we want to break them apart, we're actually going to insert a water molecule, insert atoms into that bond, and that is what's going to break them apart, and that is a hydrolysis reaction. Now, our lipids, these are our fatty acids. These are going to be primarily hydrogen and carbon atoms all linked together. However, there are going to be some variations on them. These are, again, for the most part, going to be non-polar covalent bonds, meaning they're going to share their electrons evenly amongst themselves. And because they're primarily hydrogen, carbon, and they have even sharing of electrons, they're hydrophobic. They do not mix well with water. Case in point, think about the last time you put some oil in water, either because you were doing the dishes or maybe you were making a vinaigrette, so it was oil and vinegar. The oil sits on top. That's hydrophobic. There are 5 classes of lipids. You may have heard of some of these, you may not have. Triglycerides, that's where we're really going to focus. Ketones, we're going to touch on them. Phospholipids, we will talk about in detail when we talk about the cell. Eicosanoids, we're not going to touch on them at all. They are a relatively new discovery. We're still figuring out exactly what they do, where they go, all that fun stuff. They are mentioned in the chapter, so if you are reading the chapter, like I hope you are, you will come across them. For your information, I do encourage you to read about them, but I'm not touching on them. They're not going to be anywhere on the exam for me. And then steroids. When I say steroids, you probably think about the drug. You think about maybe corticosteroids or the drugs that like WWE wrestlers and people like that take. That's not what we're talking about. We're talking about the cholesterol-based compounds that our body makes so we can run, so we can function. A variety of our hormones, as we'll touch on once we get there, are steroid-based. Steroids in and of themselves are not harmful. It's their exact compound, how they're used, and the dosing. There's a saying, the dose makes the poison. That is completely applicable here. Something can be completely beneficial in small doses, but become adverse with large doses, and vice versa, depending on what it is. So just bear that in mind. All right, triglycerides. Here we have, I will have a picture in a moment of a triglyceride. Again, going back to medical terminology, tri means 3. So we have 3 of something. And in this case, the 3 is going to be a fatty acid, or fatty acid chain. So when we have a triglyceride, we have a glycerol at the beginning. A glycerol is a 3-carbon alcohol group, and attached to that glycerol, we will have a chain of carbons. Excuse me, it's actually carbons and hydrogens, and you'll see that in just a second. And these chains will attach to the glycerol. 3 chains per glycerol if we're talking about a triglyceride. And how those carbons are linked together and whether or not they are maxed out on the number of hydrogens that can attach to them will determine if it's saturated or unsaturated. Saturated means every single carbon has 2 hydrogens, completely maxed out bonds. If it is unsaturated, then they are not completely maxed out and at least 2 carbons are double bonded together. Could be more. If we are talking about unsaturated fatty acids, we then need to talk about whether or not they're trans or cis. In other words, the carbons that are double bonded together, what sides are there one hydrogen attached to? Are they on the same side or opposite sides? I have pictures that go along with this. We'll get to them in one second. These fatty acid chains are nonpolar, meaning they share equally, and again, hydrophobic. They don't like to mix with water. Here is our glycerol group, and here are some examples of these fatty acid chains. This one right here is a saturated. Every single carbon has 2 hydrogens. We're ignoring this first one here because it's going to attach to the glycerol, so we're going to ignore that for the moment. This first one for the moment. So this is saturated. Every single carbon has 2 hydrogens, no double bonds, maxed out. These 2 right here are going to be unsaturated. You can see that not every single carbon is maxed out. We have a double bond right here, so 2 carbons are sharing 2 electrons instead of 1. And this middle chain right here is an example of a cis transformation, or a cis formation, meaning the hydrogens are on the same side. The hydrogens that are attached to the carbons involved in the double bond, they're on the same side. And this one is an example of a transformation. The hydrogens attached to the carbons involved in the double bond are on opposite sides. And you can flip which one is on bottom and which one is on top. It is still going to be referred to as a trans fatty acid. Now, all of these fatty acids have names, and those names are dependent on what makes one fatty acid a oleic versus an aldehydic is the number of carbons. These carbon chains can be anywhere from like 6 to something like 40 different carbons, like linked together. So it can be really, really short or really, really long. So this is an example of that all put together. This is a triglyceride. We have our glycerol group here, and then we have our 3 carbon tails attached to that glycerol group. Ketones. Now, ketones are going to be important for our friends who are diabetic and for anybody on a low carbohydrate diet. So the carnivore diet or the keto diet or an Atkins diet, South Beach if that's still a thing, any of those low to no carb diets. Basically what happens is these triglycerides are broken down in the adipose tissue. And when they are broken down, they are chunked into sections of these tails, okay? So we chunk off 2 carbons, we chunk off another 2 carbons, we chunk off another 2 carbons, another 2, another 2. And we're going to do this through hydrolysis. So we're going to insert a water molecule in between the bond, or in the bond between 2 carbons, removing 2 carbon chunk. Now these fatty acid chunks, These carbon chunks are going to float through the bloodstream, and eventually they are going to come to the liver. The liver is then going to convert them into ketones. What is a ketone? It is an acetoacetic acid, an acetone, or an alpha hydroxybutyric acid. I think I have pictures. No, I don't. So basically, it takes those two carbon chunks with their associated oxygen and water molecules and converts them into a slightly different formation. And then that converted formation can then go into our ATP production cycle, our glycolysis Krebs electron transport chain. Here's the thing. This takes time. And them going into that ATP production process also takes time. So if this is my primary energy source, we're going to have a ton of fatty acids floating through the bloodstream. This can lead to buildup of the fatty acids and fatty plaques that line our cardiovascular system, a condition known as atherosclerosis. And the ketones themselves can also build up, leading to a condition known as ketosis or ketoacidosis. This is typically, you can notice somebody who has it because they have a very fruity or like alcohol, acetone-like smelling breath. If you are the person in question, when you go to the bathroom, your urine will have a more fruity, alcohol-y type smell to it. Because your body's like, I have all these ketones, I can't use them. You can't use them fast enough. They're actually kind of harmful when they build up. So I need to get rid of them. How do I get rid of them? I exhale them through my breath and I excrete them through my urine. You also get rid of some through stool, but those are the two primary processes. So this is why that occurs. Your body is trying to use energy, but it can only use so many of these at a time. Phospholipids, we're going to talk about them more with a cell, because phospholipids are what make up a cell wall. So phospholipids are similar to a triglyceride, except for the fact that that glycerol group is slightly modified. And the tails are also modified, in the fact that instead of 3 tails, you have 2. This modification to the head and tail complex actually leads to a polar head and a non-polar tail. So we still have that hydrophobic, water-hating carbon chain tails, I should say. But the head, the glycerol group, and the other new addition, they are actually going to be polar, so they're going to share their electrons unevenly, and they're going to have a greater bond with water. And it looks like this. So we still have 2 carbon tails, we still have a glycerol group, However, on the other side of the glycerol group, opposite where that 3rd tail would be, we're going to have a phosphate-containing group. And it's this phosphate-containing group that is unevenly sharing electrons and has that water-loving aspect. It likes to hang out, bond with, mix with water. Here's an artistic rendering of that. You've probably seen this in previous classes, other textbooks. Very classic representation. And because of this one side likes water, one side doesn't like water aspect of phospholipids, this can create unique structures. Basically what's going to happen is when you have a bunch of these phospholipids, they're always going to try and keep their tails away from water and their water-loving heads towards the water. So they're going to turn the tails towards each other and the heads out. This can present in a couple of different ways. If we have enough, we can get a phospholipid bilayer. This is what we're going to talk about when we talk about a cell wall, when we talk about the cell. It is 2 layers of these phospholipids with the tails pointed towards each other and the heads on the outside. Or they can form a sphere, and in that sphere, all the tails will be pointing towards each other, and the heads are all lining the outside. This is known as a micelle. Phospholipids in humans also form surfactant. We'll talk more about surfactant with the respiratory system, but surfactant is the detergent, so to speak, that helps break up and maintain the perfect amount of water tension so that your lungs don't collapse, but they're still able to move so that they can expand and contract as they need to. So this is the miracle that we discovered for preemies. We can actually create this artificially and inject it until preemies are able to make it on their own. It saves so many lives. Moving on to steroids. As I mentioned in passing, steroids are all derived from cholesterol. Now, again, cholesterol gets this bad rap. Oh, cholesterol's evil, cholesterol's bad. No, cholesterol is good in moderation, in the proper amounts, in its homeostatic equilibrium. In and of itself, it's not bad. Now, cholesterol does a lot. We're going to talk about it when it comes to the cell wall, but we're going to focus more here when it comes to hormones. Steroid hormones are things like estrogen, progesterone, testosterone, aldosterone. These are all steroid-based hormones and therefore cholesterol-based hormones. So to go back to, cholesterol is good in the proper amounts. If you have too much cholesterol, one of the ways it may manifest, depending on the other processes in your body, is you may have too much estrogen or you may have too much testosterone. Or, on the flip side, if you don't have enough cholesterol to do everything that your body needs, you may not have enough estrogen. You may not have enough testosterone. So, this is some of that complexity that comes into the fact that we are a living, breathing, complex organism. Nothing stands alone. Everything feeds into itself. So what do these look like? Well, this is an example of a cholesterol. It is a linking of several hexagon rings and a pentagon, and there's various groups attached to it. In fact, we have a nice long carbon chain here. Now, how do we modify this? Well, we can modify the appendages, we can modify where the double bonds are, we can add or remove carbon chains. But this basic 3 hexagon, 1 pentagon linking is our steroid foundation. And it all comes from this. So this right here is an example of what a testosterone looks like. Moving to proteins. Proteins are let's talk nomenclature here. When I say protein, I am actually talking about a linkage of monomers. Proteins are actually going to be a polymer or chain. So whenever I say protein, I'm actually talking about a chain of amino acids. Amino acids are actually the monomer or building block of our protein complexes. In humans, we have 20 different kinds of amino acids, and these are our building blocks. Everything in us is made from these 20 amino acids. Now, from a nutrition standpoint, you may have heard of essential amino acids. There are 9 of those bad boys, and what that refers to is the fact that we cannot make them. So of these 20 that we use, 9 we have to get from our diet. We can make the other 11 through various processes and the encoding within our DNA. The other 9 I have to get from my diet, which is why with our vegetarian friends, we strongly encourage seeing a nutritionist, because it's very easy for them, if they're not careful, to miss some of those essentials, especially if they're not eating a variety of protein sources. Maybe they're just relying on tofu versus eating tofu and like 3 or 4 different types of beans. So that is one of the reasons we encourage always seeing a nutritionist when you're trying to go on a vegetarian or vegan diet. making sure you're not missing any of those amino acids. Here's an example of an amino acid. This is the base formation of an amino acid. Every amino acid is going to have a nitrogen-based amino group on one side. On the other side is going to be a carboxyl group, which is a hydroxyl with a carbon and a double bonded oxygen. Every one of them is going to have that. What makes every amino acid different is the R group. So coming off of the center carbon will be what we refer to as the R group. The R group is what changes, and it could be almost anything. For instance, this alanine right here is a carbon hydrogen complex. A single carbon with 3 hydrogens. That's it. Versus this one, which is a carbon with a ring and a hydroxyl group. This is tyrosine. This one is a carbon chain with, again, oxygens attached at the end. This is a glutamate. Versus this lysine, which is a carbon chain that terminates in another amine group. It's a nitrogen group. These amino acids are actually classified into various categories based upon what their R group is. Does that R group contain a ring? Then it's this type of amino acid. Is it just a carbon tail? Then it's this type. Does it contain a nitrogen? Then it's this type. If you end up taking a biochemistry class based on the degrees that you're going for, you will have to learn these. Most biochem teachers make you learn the groups and what belongs to what group. So just a fair warning on that one. How heavily, though, that does vary depending on the biochem teacher. So just be aware of that. We're not going to focus on that in this class. Just be aware that the R group is what is different. All right, now, amino acids link together and form what we call peptide bonds. Peptide bonds are a covalent bond in which the carboxyl group on one amino acid and the amine group on another amino acid have been joined together. Peptides, again going to nomenclature, are going to be linkages of either 2 to 50 amino acids. Anything over 50 is what we refer to as a protein. Amino acids, when they bond, have levels of structure. So that primary linking, this right here, this initial formation of a peptide bond, that is known as primary protein structure. That's the first level. And then, once they've linked together, we're going to get hydrogen bonding between the amine hydrogen on one amino acid and the carboxyl oxygen on another amino acid, but they're not next to each other. So we're going to get additional bonding, but this is just hydrogen bonding, so it's much weaker, between non-adjacent amino acids. And this is going to form the secondary protein structures. This is going to be traditionally in the form of like alpha helixes and beta pleated sheets. This would be an alpha helix. You can see the hydrogen bonds forming between non-adjacent amino acids, and this is pleated sheets. Again, hydrogen bonding between non-adjacent amino acids. Then we move into tertiary. Tertiary, we're going to get bonding between R groups. So this is the R groups are going to start to interact together and form additional bonds. This is going to form a whole bunch of bends and loops in that protein chain, and looks something like this. This is an alpha helix and a beta pleated sheet, but we're going to get all sorts of wonkiness going on. That's tertiary. Some of the interactions, so some of the bonds that are going to form in this tertiary structure are things like ionic bonds, van der Waals forces, hydrogen bonds, covalent bonds, especially if we have sulfur involved. Some of the R groups do have a sulfur, and they will form what's referred to as a disulfide bridge. So lots of different bonds involved here. The breaking of any of these bonds, whether it's the primary peptide bond, the secondary hydrogen bond, or any of these tertiary bonds, is referred to as denaturation. So if you hear about people talking about denaturing your proteins, all that's referring to is the breaking of those bonds, which is what has to happen if we want to reuse those amino acids in any other way. Last but not least, we have quaternary protein structure. Quaternary protein structure is simply we take one protein chain and another protein chain and we bond them together. So this is the formation of proteins with more than one polypeptide chain. Things like hemoglobin. Hemoglobins are actually 4 protein chains that have been bonded together. And located in the middle of all of those protein chains, or each of those protein chains, I should say, will be a heme group. Moving to our nucleotides. Nucleotides and nucleic acids. Nucleotides have a basic structure just like amines do, or amino acids do, I should say. They are going to have a phosphate group that is attached to a 5-carbon carbohydrate that is then attached to a base containing a carbon-nitrogen ring. Now, here's where we get some of our variations. That 5-carbon carbohydrate is either going to be in the form of a ribose or deoxyribose format. I'll show pictures in just a second. And the base containing carbon-nitrogen ring will be in the format of either a pyrimidine base or a purine base. Pyrimidines are going to come in the form of cytosine, thymine, or uracil. Purines are adenine, guanine. And if those are ringing a bell because of genetics, that's right where we're going. So here we have, this is a deoxyribose, and this is a ribose. They're both a pentose, but a deoxyribose only has one hydroxyl group down here. Actually, technically it's got 2 because there's one up here. This ribose has 3, 2 down here and 1 up here. So that's the difference, is an extra hydroxyl group. Then they can attach to one of these carbon-nitrogen bases. Pyrimidines are going to be a single hexagon ring with a combination of hydrogen, nitrogen, carbon, and oxygen in various formats. So you can see a cytosine versus a thiamine versus a uracil. And then purines are going to be a pentagram attached to a hexagon ring. So 2 rings, and that's going to be our adenine versus our guanine, but again, it's going to be a carbon, nitrogen, hydrogen, oxygen complex. And that's the main difference. A lot of the differences in our molecules are simply where things attach. Do they attach on the first carbon versus the second carbon? And hopefully you guys are recalling that from your chemistry classes. All right, now, under the nucleotide heading, obviously we're talking DNA. But we're also going to be talking about energy transferring nucleotides. Things like ATP, adenosine triphosphate, or ADP, adenosine diphosphate, 2 versus 3 phosphate groups. Those are actually going to be nucleotide based as well. It's an adenine, so this guy right here, attached to a ribose, this guy right here, and then 2 to 3 phosphates attached as well. So instead of this phosphate group, or just one phosphate group I should say, in order to make it a ADP, there'd be a second one right here. So we'd have adenine right here, ribose, 1 phosphate, 2 phosphate because it's ADP, or if it's ATP, there would be a third one right here. So it's a variation on our genetic code, or genetic coding material, I should say. We also have nicotinamide adenine dinucleotide. Again, that's going to be a variation on our complex right here, and flavin adenine dinucleotide. nicotinamide adenine dinucleotide is NAD, and a flavin adenine dinucleotide is FAD. We will mention these again when we talk about energy transference. All right, then we have at that phosphate group, it can actually form a ring, and these are referred to as cyclic nucleotides. They're typically messenger molecules, so we will mention them in passing, but we're really not going to spend much time on them or their formation and shape. But if you hear things like CAMP and CGMP, these are cyclic nucleotides. It is, again, this complex, only this phosphate group is a ring as well. And that's a messenger molecule. So our traditional view of nucleotides is going to be for our genetic code, our expression and storing of how we are going to make various things and present them. So DNA is going to be that storage aspect. RNA is what's going to actually allow for the expression. DNA is going to be the deoxyribose ring and can use either adenine, guanine, cytosine, or thymine bases. It is going to present in a double-stranded helix, so it's going to be a spiral with 2 strands, and hopefully this is something you remember, but it's going to undergo a law of base pairing, meaning adenine is always going to form a bond with the thymine, and cytosine is always going to form a bond with guanine. So if I have a cytosine on one side of the spiral, the other side needs to be a guanine. If I have an adenine on one side of the spiral, the other side has to be a thymine. Now, right here though, you see a uracil. Where's that come into play? That's going to come into play when we are copying our DNA. And when we copy our DNA, RNA does not use the thymine base. Instead, it uses the uracil. So when we copy and we're pairing, doing a complementary pairing with RNA, any place we would have put a thymine opposite an adenine, we're actually going to put a uracil instead. It looks something like this. Very classic representation right here. RNA is going to use the ribose base and is going to be able to use the adenine, guanine, cytosine, and uracil bases. All right. Moving into our cells, actually. So, we have this lovely thing called differentiation. We have over 100 trillion cells in our body. They are all derived from one sperm, meaning one egg, and one set of DNA. Same basic components, but they all form 100 trillion. Those 100 trillion cells are going to be divided into 4 general categories, but we have so far discovered about 200 different types of cells. And the reason, even though they all start the same, they all came from one egg meets one sperm, and then they just divided and divided and divided, is something called differentiation during development. Basically, during development, different markers were turned on and off in the genetic code, which changed the cells. So instead of becoming a skin cell, it became a muscle cell. Instead of becoming a muscle cell, it became a bone cell. And we're not going to get into that in this class. That is an entire semester worth of conversation. We do touch on it in our zoology class, though, so if you took that, you will have had some of that already. We do touch on it. There's another class we touch on it, but not here. I'm spacing the other class. Basically, because one cell is next to another cell, it will turn certain markers on, certain markers off. If certain chemicals, if they're exposed to certain chemicals, hormones specifically, then it will turn certain markers on, certain markers off. So it's super, super complex, and it's one of the things that they are still studying because it's where some of our genetic abnormalities come from. So that's one of the things they're looking at. We're not going to get into that. That is at least an entire semester in and of itself to really start to understand it. All right, so in our normal, regular cell, generic cell, every cell is going to have a plasma membrane or a cell membrane. The terms are kind of used interchangeably, and I will truthfully probably stick more with cell membrane. Basically, it is a barrier between the inside of the cell and the outside of the cell. And the fluid inside of the cell is going to be referred to as intracellular fluid. We mentioned that with our fluid compartments in chapter 1, and that is abbreviated ICF. And the fluid outside of the cell, as we mentioned, is extracellular fluid, or ECF. Inside the cell, it is going to be divided into various components. We are going to have our nucleus, and we're going to have our cytoplasm. The cytoplasm is further divided into cytosol and organelles. So basically, when we're referring to cytoplasm, we're talking about basically everything inside the cell except the control center. If we're just talking about the fluid of the cell, we really should be using the term cytosol. Cytoplasm is the fluid plus organelles. And cytosol is just the fluid. So this is an artistic rendering of a quote-unquote typical cell. We have our cell wall, and then we have a cutaway showing a mitochondria over here. Here's our nucleus with our nucleolus. Here's our smooth ER and our rough ER. Actually, this is all rough. This is smooth over here. Here's our Golgi apparatus, our centrioles, our peroxisomes and primary lysosomes. Lots of structures. We're going to touch on some of these. All right. Plasma membrane. We're going to start there. So the plasma membrane is also referred to as a fluid mosaic model, meaning it is a phospholipid bilayer that has embedded within it and on it a variety of cholesterols, membrane proteins, and membrane carbohydrates that allow it to maintain its shape, accommodate various substances, and allow certain things to come in and certain things to come out. Selectively permeable. This is an artistic rendering of these structures. Here we have a phospholipid membrane. So phospholipid bilayer. So we have two of them. And then embedded within it, we have integral membrane proteins, also sometimes referred to as transmembrane proteins. These are proteins that are going to completely go through the phospholipid bilayer and have a component of it on the outside of the cell and a component of it inside the cell. These can be a variety of different things. They can be channels, they can be receptors, they can do a bunch of different things. Then we are going to have integrals that only go partially in. So this is why we sometimes have that different nomenclature. Transmembranes are always going to be the ones that go all the way through. Integrals can also refer to ones that just are embedded into. So they only go partially through. Then we have peripheral membrane proteins, and peripheral membrane proteins can be either on the inner layer, and they just sit on the surface, or they can just sit on the surface on the outer layer. Most of peripheral proteins are going to be found attached to the inner layer, and the reason for that is that the cytoskeleton of a cell, the fibers that give it its shape and structure, anchor to the peripheral membrane proteins attached to the inside membrane, and so most of them are on the inside. We're also going to have embedded into the cell membrane, we're going to have cholesterol molecules. We'll mention more about them in a second. We are going to have carbohydrate chains that are attached to proteins, and we're going to have carbohydrate chains that are attached to the phospholipids themselves. These carbohydrate chains are going to act as markers. If you've heard of cell markers, that's the carbohydrate chain. This is what is telling a white blood cell that this does or does not belong. Mentioned this already, most of this, however, we have not talked about this. So, we talk about water as if it's able to pass easily through this cell membrane. It can do whatever it wants and flow easily in and out. True, but not true. Water cannot get past the phospholipids. Because of that hydrophobic tail component, water cannot slip through between the molecules. So what's embedded in the cell membrane are channels that only allow water through, and they're referred to as aquaporins. And they're always open, and they allow water to move through the membrane easily. But water itself cannot get past the phospholipids unless it's coming through this channel. And this channel is going to be an integral membrane protein, or a transmembrane protein, to be specific. Cholesterol. Cholesterol is going to add an aid in the fluidity of the cell membrane. It's actually also going to decrease the permeability of the membrane to water, making it even more difficult for water to get past, unless it's going through the channel. Basically, what cholesterol does is it interferes with some of those hydrophobic interactions between the phospholipids, preventing the phospholipids from crystallizing and clumping up. So basically, cholesterols are going to help maintain the sheet-like formation. All right, so if we wanted to figure out how much of the cell membrane was protein versus fatty acid, we would actually have to disrupt the membrane. We would have to do an experiment and physically remove the proteins, which would completely cause the phospholipid membrane to kind of fall apart because they are integral. They are embedded within. These membranes are going to have both hydrophilic and hydrophobic parts, so they're going to be water-loving and, water-hating, because they have to be able to talk to both the water-loving and water-hating components of the phospholipids. And the fact that they can be loving and hating makes them amphipathic. As I mentioned, some of these transmembrane proteins are going to be channels or carriers. If they're only located on one side, these are actually going to be more of our enzymes and receptors. I actually misspoke just 2 seconds ago. If they're peripheral membrane proteins, they are going to be easily disassociated from the cell membrane, so they can become easily detached. As I mentioned, most are located on the cell side or the inner layer, and they are going to be associated and involved with the cytoskeleton. Membrane carbohydrates are going to be covalently bound carbohydrate chains to either lipids or proteins, so glycoproteins, glycolipids, and these are going to be involved in things like glycocalyx, but far more importantly and commonly, cell recognition. Do you belong? Let's talk about the nucleus. So the nucleus is going to be composed of an envelope or a membrane. Embedded within that membrane are going to be pores, and these pores are going to allow things to move in and out of the nucleus. We're also going to have a section of it that is going to be the nucleolus, and the nucleolus is the site of ribosomal RNA synthesis, so rRNA synthesis. This ribosomal RNA is going to be important for us to make proteins. So the function of the nucleus is going to be to contain all of the DNA within the cell except for a small portion in one location. DNA, remember, stores genetic code, and then that DNA is going to be transcribed or read to form a single RNA chain, and that RNA chain is going to allow us to express or utilize that genetic code. And that RNA chain is going to leave the nucleus to go do its thing wherever it needs to do so. Kind of speaking generically, because to get specific, we've really got to get specific, and then the unique instances, it can be here, 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 and we just kind of bog down. This is just an overview, a refresher, because you have had this in theory in previous courses. We talk about the cell in Gen Bio, we talk about it in zoology, we talk about it in anatomy, we talk about it in microbiology. You've had this before. So this is supposed to be just a refresher. Cytosol. As I mentioned, the cytosol is the fluid component of the cell, and cytoplasm is the cytosol plus the organelles. So the cytosol is going to be the location of some very specific chemical reactions. We're actually going to mention one of them. It's also going to be the location for the storage of fat and carbohydrates in the form of inclusions. Inclusions are going to be basically like little pockets. So they don't necessarily have a cell membrane. They're not enclosed by anything. They're just little balls of fat and carbs. And this is also where we're going to store our secretory vesicles, which do again have membranes. Organelles are divided into membrane-bound and non-membrane-bound organelles, so membranous and non-membranous. Membrane-bound organelles are going to be organelles that have a membrane that separate them from the cytosol. It's kind of self-explanatory, hopefully. And these include the endoplasmic reticulum, both smooth and rough, Golgi apparati, lysosomes, peroxisomes, mitochondria. All of these are going to be membrane-bound organelles. So if you have a quiz or a test question asking you what lists the membrane-bound organelles, that appears somewhere on one of the exams. It's either the final or the first exam or both. If you have a question that says, what's a membrane organelle, and list me and tell me what they do, this is the slide you go to. Slide number 50. Endoplasmic reticulum is divided into two categories, smooth and rough ER. Smooth ER is going to be the endoplasmic reticulum that is going to be responsible for forming or in creating carbohydrates and fatty acids. Rough ER is going to be the endoplasmic reticulum that is responsible for forming proteins. Now, rough ER is going to have an appearance that it has flattened sacs and very bumpy or granular, hence its rough ER name. The reason it's going to appear granular or bumpy is due to the fact that the membrane is embedded with ribosomes, which is what enables the rough ER to create proteins. The smooth ER is going to be tubular in nature. It's going to have a smooth appearance. And the smooth and rough ER, if a cell has both, they're going to run continuous with each other. So they're going to run from one right to the other. There's not going to be a break. Now, not every cell has rough ER, and not every cell has smooth ER. Because not every cell creates fatty acids and carbs, and not every cell creates amino acids. So, bear that in mind. Artistic rendering of what that looks like. The rough ER or the smooth ER, if there is no rough ER, is going to be attached to the nuclear envelope at a nuclear pore. So the RNA is going to come out of the nucleus straight into the ER. This is the rough, this is the smooth. Rough ER, as I mentioned, packages and synthesizes proteins. This is going to allow for them to go to certain organelles within the cell. They can be exported out of the cell. They can be incorporated into the plasma membrane, whatever is needed from this protein. Smooth ER is going to be involved in particularly fatty acid synthesis, especially triglycerides and steroids. However, it also stores calcium. And this is going to be really, 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 I cannot express how important this information is. Smoothie R stores calcium. This is critical in our muscles. So when we get in module 3 and we are talking about skeletal muscle and skeletal muscle contraction, and then we move into the cardiovascular system and we are talking about heart contractions, calcium and therefore the Smoothie R is going to play a critical role in proper function of these mechanisms. So we will come back to the fact that Smoothie R stores calcium in those chapters, but heads up, it's coming and it's important. In some cells, Smoothie R is specialized. For instance, in the liver, Smoothie R is actually where the detoxification enzymes are located, and so that is why liver cells are able to detoxify. What do they detoxify? They do things like break down alcohol, drugs, medications. All that's because of the Smoothie R specialization. Golgi apparati. The Golgi apparatus looks like a stack of flattened pancakes, so to speak. It has the ability to do the finishing, polishing process to proteins. Basically what it does is a protein comes in and it will get packaged up, snipped, cut off, things that don't belong, removed, stuff like that, and then final packaging and usually sent out of the cell. Golgi apparatus can also package things like carbs and fatty acids as well, but we typically talk about it from a protein aspect. Think of it like the post office. You might have an object, but maybe you don't have a box, or maybe you don't have an envelope, you don't have a stamp. You go to the post office, you get the box, you get the envelope, you get the stamp, you put your thing in the envelope, you put the stamp on it, and then you give it to them and they send it to wherever it needs to go. That's what the Golgi apparatus does. It's the post office of the cell. It looks like this. It has a cis face and a trans face. Why one is called cis and one's trans, that I actually do not know. There's probably a reason, but I am not aware of it. But the cis face is going to be this aspect of the Golgi apparatus that is facing the nucleus. The trans face is going to be the aspect of the Golgi apparatus that is facing the cell membrane. Mitochondria. We're going to spend more time with mitochondria when we talk about cell metabolism. Mitochondria are the powerhouses of the cell. This is where we're going to have the Krebs cycle that's going to occur in the matrix of the mitochondria. We're going to have the electron transport chain occur across the cristae of the inner membrane. This is where we generate the vast majority of our energy. Mitochondria are unique. They are the only organelle in the cell, other than the nucleus, that has DNA. In fact, they have a little ring of DNA. It's referred to as a plasmid. And this DNA is distinct from the nucleus. So it is completely different. There's a couple of scientific theories that have the mitochondria being a symbiotic organism back in the day. Basically, we had our cell, or a eukaryotic cell, which is what we are, and it formed a symbiotic relationship with a bacteria or another cell, but they're thinking bacteria, and because of that relationship, eventually the bacteria came into the cell and it integrated itself so completely that it lost its uniqueness and ability to thrive on its own and became what we know today as mitochondria. It's a theory, but it's an interesting theory. There are lots and lots of research papers. If you're interested in learning about that theory, lots of research papers out there for you. But that is why it has a ring, we think. If you are interested in how things are related to each other, you'll have heard zoologists specifically talk about mitochondrial DNA. That's where it comes from. This is an artistic rendering of a mitochondria. Here we have our outer membrane. Then we have our inner membrane that forms folds. These folds are the cristae. And the fluid within the inner membrane is the matrix. So when we talk about energy production, and I mentioned the cristae or the matrix of the mitochondria, this is what I want you to refer back to. We're going to pause here, and you're going to want to go to the second audio.